Hello, I'm talking to you. This is Goddess of Science Biology, and I'm Mrs. Bolden, and today we're going to solve the mystery of how we get 4 bases to code for 20 amino acids in part 8 of my molecular genetics series. Okay, let's go. So, why do 4 bases code for 20 different amino acids? That's kind of like a weird, like, why don't we have 20 different bases? And each one could code for a different amino acid, but that's not exactly how it happens. So let's dig into this. So there's a little bit of math ahead. Just be careful. I'm with you. Don't worry. It'll be okay. So if one base coded for an amino acid, well, that would be adenine would code for amino acid, thymine amino acid, that's 2 amino acids, guanine would be an amino acid, would code for an amino acid, and cytosine would code. So we would actually only get 4 different amino acids, but we know that there are 20 different amino acids. So it couldn't be a one-to-one correspondence there. So there's not enough if we just have one base as one amino acid. So if you have 2 bases coding for an amino acid, that means that you could have all of these different combinations, and we would actually have 16 different combinations. But that's still not enough because we have 20 different amino acids. So the number of combinations are the number of things that we have, 4 different bases, and we're going to take them 2 at a time. So it's 4 squared, and 4 squared is 16 different amino acid codes. Still not enough. So let's try 3. So if you have 3 bases coding for an amino acid, that would mean it would be 4, because we still have 4 bases, taken 3 at a time. So 4 cubed would be 16 times 4 is 64. So 4 cubed combos are 64 amino acid codes. Hey, now we have enough. We didn't have quite enough when we took 2 bases coding for an amino acid. So now we have, if we take 3 bases coding for an amino acid, we're going to have more than enough codes, which is okay, because sometimes if you have 2 codes that have 2 of the amino acids the same and a third that is different, they can still code for the same amino acid, which is kind of good, because if there's a change in the one amino acid that's different, it may not code for a different amino acid. So a codon, they figured out, is 3 bases that code for an amino acid. And all the time when I had you guys converting DNA to DNA and DNA to RNA, I always put them in groups of 3 in preparation for this, because that's how the DNA is read by the machinery that makes the RNA and also makes the protein. So here's the thing, most amino acids are coded for by more than one codon, and this actually works out good because sometimes you can have like a change in one of the bases, but it doesn't necessarily code for a different amino acid. And if that happens, the protein is not going to be changed, because if it normally codes for alanine and you change one amino acid and it still codes for alanine, it doesn't matter. It's just, that's kind of like a little safety feature that we have in our DNA. There are some exceptions in mRNA, that there are some special codons. The AUG codon codes for an amino acid called methionine. Methionine is the very first amino acid in all proteins. Sometimes the methionine gets snipped off in post-production, but sometimes it stays there. So AUG, besides coding for methionine, it also is the start codon. So this is kind of the signal that says, hey, it's time to start the codon, time to start coding here. This is the very first one. So that is, okay, where's the next one? So if we have a start codon, we also must have to have a stop codon. And we have three different stop codons, UAA, UGA, and UAG. All three of those are stop codons. So the machinery starts where it gets an AUG. It reads an AUG. That's when it starts. And then when it gets to one of the three stop codons, the little ribosome lets go, and it knows that you should not continue any further. These three don't code for an amino acid at all. So here is the secret agent RNA decoder chart. So the way that you use this thing, and I have a different version if you're not really excited about this, and I'll probably hand one of these out to you in class. So when you have a codon like AUG, you start in the lightest part in the middle. So the first base is A, and then for the second base, you go to the next ring out, which would be here, AU, and then you find the third ring is the third nitrogen base. So AUG is, in fact, methionine. You could do CCC, so C, and then we go to the next ring is C, and then C is proline. Now notice all three different bases here in the third position with CC all code for exactly the same thing. So we could swap a C in the third position for a U, and it would not change the amino acid that it's being coded for, which is kind of nice. Here's our stop codon, one of our stop codons up here, UGA, and then the other one is UAA or UAG. So those are stop codons. Everything else has its own code, and sometimes there are four, sometimes there's not as many, just kind of depends, but they all get used for something. So this is a secret agent mRNA decoder chart. Notice there aren't any T's on this because there's no T's in RNA. So you have to change the DNA to RNA to be able to use this chart. Here's the other version of the chart, and the way you use this one is the first base, you look down this side. So if it was a C, so if we're going to find CCC, so then we're going to look in all of these bases, and then we're going to come over to the second base, which is across the top. So now we know it's one of these three. And then to figure out which of these three, we go over here and follow that line over to our box that were possible. So CCC is in fact proline. This one, it works okay. It's a little easier to use the circle one for me, but if you enjoy using this one, just let me know. I can supply you with this chart as well. Okay, that's it for this time. Thanks for joining me. Have a great day.